this is Eric Topol, and this is a new Medscape podcast, Medicine the Machine, and I'm fortunate to be with my co-host, Abraham Verghese. And we welcome today Professor Shane Crotty. Shane has a remarkable background, having gone to MIT, UCSF, Emory Vaccine Center, and then landed here in La Jolla, where I live, but works at a different institute, La Jolla Immunology Institute. He's been there for about 18 years now. And you wouldn't know it because he's kind of has a baby face and looks like a young guy. Anyway, Shane, welcome. It's really great to have you.
Thanks for having me. Yeah, I started young.
So the reason we wanted to get Shane with us today is he has been, if you are active on Twitter and if you read the major journals on COVID, you can't escape that Shane is there, whether it's in Science magazine or Cell or you name it. And on Twitter, he's a great educator about immunology and virology. So I guess I want to start, Shane. You took on a big mission here to try to educate the social media world beyond the normal academics, right? I mean, writing papers in the top tier journals. And I want to get your thoughts about the challenge of basically the medical community that's Medscape isn't necessarily, the experts in immunology, but you are. So maybe you can address kind of this mission you've been on.
Yeah, sure. I didn't do much on Twitter for a long while. Science Twitter is pretty great, particularly during COVID-19, because there's a lot of really good filtering for good preprints, and even some people are even just publishing data right on Twitter before they had it anywhere else. So it's definitely had some real scientific value. But yeah, the public education part of it is different and definitely harder. I guess I've always found communication to be something I felt important to the public. Actually, at MIT, I double majored in biology and in writing, and the writing part was in the interest of communicating to non-scientists. And so in COVID, for me, actually, that really, I think, came to a head when the Pfizer and Moderna clinical trial data were available, but they didn't yet have that emergency use authorization, right? So right around Thanksgiving. And so these vaccines looked incredible, but a lot of what was in the news and a lot of what was on Twitter was about fear of RNA vaccines. And at the time, they were being called genetic vaccines a lot, right? And I didn't see any correction to that, right? Or better communication of it. So I think I'd gone to work for about 200 days straight at that point since March. So I was taken off Thanksgiving Day, and then I decided, oh, well, why don't I try and write something for Twitter on RNA vaccine safety, and what an RNA vaccine actually is and how it works, and that actually the safety data in those clinical trials was phenomenal. And so that's what I did, and it got seen like 1 million times in the first 24 hours, and like 3 million times in 72 hours. And that's what I referred to RNA as being like a post-it note, right? It's just this temporary message that the cell writes instructions on, and there are like 5,000 post-it notes in every single cell, and then they get shredded up, and put away. It's not like this permanent change to the cell. And then those things got used in newspaper articles and TV articles all over the place. So that's what swung it for me. And then trying to do things after that were educational, even though, as I think you were noting, the immunology gets really complicated really fast. So it's not so easy sometimes.
I must say, when you said post-it note, I mean, that's exactly the kind of colorful metaphors I think that not just the public, but all of us need to truly understand immunology, given that we did it so long ago. I remember the first time someone described an activated immune cell as being the difference between a Piper plane and a F-16. It was the moment that I understood what activation really means. So I think you've done a very nice job of explaining immunology in terms that all of us can understand. Yeah.
Well, I think, Shane, your communication at that juncture was quite important. You then reviewed the mRNA vaccines preceding the current ones and came with some grounding. But even today, there are people out there that are still trying to make this into gene therapy. There's this misinformation that has been propagated that the mRNA, goes into the brain and causes headaches and brain fog. And, it's just endless in terms of the misinformation. So we're thankful to have you trying to set it straight. And I thought what we could start with today is, a little quick review about the, interferon side, the innate immunity and adaptive immunity, the antibody B T cell side, as to when you get COVID, the sequence of events, just to kind of a little review before we get into more details.
Sure. So the model I work from is the simplified model. Is a couple of things. One is that in terms of protective immunity, the pathogenesis of the virus always matters. And so to me, it's really an infection in two stages. One's that upper respiratory tract and oral infection. And then the second is the lung infection, which clearly the lung infection and the pneumonia, that's where the hospitalizations and deaths are. And it's a different immunological battle in those two places. But then temporally, you get infected probably first in the nasal passages, and then quickly rest of the upper respiratory tract and oral mucosa. And any virus that can cause disease in humans has to have at least one good immune evasion trick. And frequently understanding that trick is important for both understanding the pathogenesis and understanding protective immunity. And for this virus, I think it's been pretty clear from pretty early on that it's one big trick, so to speak, or its biggest trick is avoiding an early type 1, type 3 interferon response, that it's just an unusually stealthy virus. And that, as a result, gives SARS-CoV-2 a bigger head start on your immune response, on your immune system than the average virus. And that's because, in general, your innate immunity is important to just dampen down the levels of viral replication and keep it from spreading further by activating those antiviral pathways in neighboring cells and tissues. But also, almost all of the adaptive immune response, that is, almost all of your antibody and T cell response, is completely dependent on that first innate immune signal. The adaptive immune response does not start until those innate immune alarm bells go off. And whether you want to think of that as a burglar alarm or a fire alarm or something like that, right? Until that alarm goes off, your B cell and T cell fire trucks are just sitting there parked because they don't do anything till they hear that alarm. And so that timing makes a big deal, right? Because the virus is replicating on an exponential scale, and then the adaptive immune response is also going to replicate on an exponential scale. So any of those time lags matter. But once you do get that response going, in an average case of COVID-19, our group with Alex Setty's group was the first to show that, if you've got people who weren't hospitalized but got symptomatic COVID-19, they make a T cell response, really both a CD4 and a CD8 and a neutralizing antibody response to the virus pretty quickly, and of a magnitude that largely fit our expectations for what, if you said, okay, there's a new respiratory viral infection that on average is causing something like cold or flu-like symptoms, how big of a T cell and antibody response, might you expect to control that? It looked in that range, and with the type of qualities that we would expect for both those T cells and B cells.
So now we're going to go fast forward. We're well into the mRNA vaccination era. Hundreds of millions of people have received them. And we're also well into Delta. And can you tell us your thoughts about how Delta is different from the ancestral strain, alpha, beta, gamma, with respect to the vaccine protection and the breakthrough infections?
Yeah, that is definitely the topic of the moment. I'd say that the quickest answer is that Delta does have partial immune evasion from antibodies. It probably doesn't have any meaningful evasion from T cells. Its antibody evasion is somewhere between the modest amount of alpha and the more severe amounts of beta. The most detailed analysis of RNA vaccine protection from Delta is really Public Health England, which calculated an 88% efficacy still against Delta for symptomatic cases, and something basically no change in protective efficacy against hospitalizations. That is, still in the 95% kind of range. That seems pretty consistent with expectations from in vitro data, from so far what's been happening in the US in terms of cases, and in terms of protective efficacy against other variants. So protection against alpha was probably only down about 5% for the RNA vaccines, whereas protection against gamma was down about 20% in sort of big study in Qatar. And so that the data from England largely look like they match expectations. There's definitely a flip side to it, right? Which is the data coming out of Israel and more concern from the Israeli government about how much protection there is against cases. And so I think we'll have to see how that plays out, but that definitely, as far as I can tell, gets pretty deep into the epidemiology of what's the proper way to calculate efficacy, and how well do you match the cases. I actually really like the commentary in the New England Journal that was with the Public Health England paper the other day about their choice of test negative controls for how they came up with the efficacy. It's conceivable there's a bit of a difference there. In England, they took this pretty bold strategy, right, as you will well recall, of giving the two doses 12 weeks apart, right, instead of 3 weeks apart. Whereas in Israel, they followed the clinical trial schedule and went with the 3 weeks apart. As a result, there are more people in Israel who are further out from their last dose, right, than there are in England. And so I think it will be interesting to see more head-to-head comparisons of that over time as it affects protective efficacy. The fact that even with pretty substantial drops in antibody titers over 6 months, and the fact that antibodies aren't quite as good against Delta as against Alpha or the original strain, that the observation that protection against hospitalizations, and by proxy deaths really haven't changed is pretty consistent with the notion that there are really a lot of different parts of the immune system that are contributing to protection at that hospitalization stage. And that really goes back to the model and thinking about this as a disease in two phases, right? If you've got the upper respiratory tract infection, but then you've got the lung infection, and they're different. And while this is a virus that replicates really fast in the upper respiratory tract, it's a pretty slow disease in the lungs. It's usually 14 days before you really run into trouble of symptoms, before you really run into trouble with that pneumonia. And it being a slow disease in that tissue means there's a lot more time for different branches of the immune system to contribute to control, whereas in the upper respiratory tract, you've only got like 3, 4, 5 days before people become symptomatic and transmitting. So while stopping that upper respiratory tract infection, and i.e. preventing symptomatic cases, is a pretty high bar, preventing hospitalization is a much lower bar, I think, because the kinetics are so different, and thus there's a lot more time for CD4 T cells, CD8 T cells, and memory B cells and recall antibody responses to all contribute to stopping the virus, i.e. stopping the virus in 14 days instead of stopping the virus in 3 days. So I think that's immunologically what the high level of protection against hospitalizations and deaths, even when protection against symptomatic cases drops for different variants.
Medicine and the Machine will be right back.
Welcome to Medscape's latest podcast series, Medicine and the Machine, featuring Medscape's editor-in-chief, Dr. Eric Topol, and master storyteller and clinician, Dr. Abraham Verghese. Remember, you can find the latest in medical news, expert perspectives, clinical tools, continuing medical education, and more at medscape.com.
In some of your other work, you have looked at administering the vaccine very slowly or over a long period of time. Other viruses that have a slow pathogenesis. Do you see us doing something different with the means by which we administer these vaccines? Not just intervals, but by the actual infusions, say, instead of inoculations.
Yeah, it's a great question. In our HIV vaccine work, we asked, do you get a different immune response if the immune system just sees the antigen over a different period of time? And so compare a single bolus injection, i.e. with a conventional syringe, or providing the antigen, same amount of antigen and adjuvant over a 14-day window instead. And the results were dramatically different. There were basically all of the monkeys made neutralizing antibodies in one case and none of them in the other, essentially. Right? And immunologically, we and Daryl Irvine at MIT, our collaborator, were interpreting that as that sort of delivery of antigen over something like a 14-day period is a lot more like what would happen during an infection. And so it matches what your immune system has essentially evolved to do. And one of the things we learned by doing that was that when you deliver that same amount of protein or same amount of antigen over a longer window of time, your immune system actually gets a lot better at retaining that antigen. And as a result, it gets a lot better at sustaining a productive immune response longer, even though in actuality, it's the same amount of material. So yeah, with those results, and it's repeated in multiple experimental systems, there's a lot of interest in trying to figure out, can that be mimicked in other contexts? The thought at the moment is that the RNA vaccines aren't doing that. The reports for the RNA vaccines have been that the RNA is probably resulting in expression of protein for just a couple of days. And if that's true, that's actually got a lot of positives on the upsides, because if that could be changed to be expression over a longer period, a week or two, that yeah, you might actually be able to get a really big improvement in the quality of the immune response just by changing the kinetics.
Well, as a follow-up to that, in your sort of widely cited Cell paper early on in the pandemic, that showed that there was a fairly robust response across all the immune system to this virus, and that there was good memory. For the benefit of people, like me, for whom Immunology 101 was a long time ago, it's interesting that we're focusing more on T cells than on B cells, when most of us think of this vaccine as in the business of generating antibodies from B cells. So explain for us again the importance of that sort of memory component, and if you would expand on that a bit.
Yeah, sure. And from a vaccine perspective, I like to frame it this way. For any pathogen, let's just say virus, that's susceptible to neutralizing antibodies, your perfect vaccine is a vaccine that elicits high levels of neutralizing antibodies forever. And you have them for, that's because if you really do that, and you can get essentially sterilizing immunity, you have such high neutralizing antibodies, you stop the virus at the front door, and that's the end of it. And so neutralizing antibodies are definitely very important. In the real world, that frequently doesn't happen. Either you can't generate that high of neutralizing antibodies, or you can't maintain them long term, or you end up with variants, right, or mutants that can get around some of that. And so that opens the questions of, okay, if the virus gets past those neutralizing antibodies, that frontline defense, do you have additional lines of defense, and or do you have redundancies and backup systems in place? And so for an acute viral infection like SARS-2, that's really where the T cells come into play, where probably once you are infected, the T cells become more important than the antibodies, because now you have a lot of virus inside of cells, generally inaccessible to the antibodies, and then it looks more important to have the T cells to clear that infection. And combined with that, it's generally very hard for acute viruses to evolve any escape to T cells, because there's so many different epitopes available for the T cells to recognize, and my T cells recognize different epitopes than your T cells. So if the virus could escape some of my T cell epitopes, they wouldn't be escaping any of yours. So there's not that much evolutionary advantage. So it makes sense for there to be a high level of attention on the antibodies, but just a recognition that they're not the only source of protection, probably, and particularly when you shift from, again, talking about protection from infection versus protection from hospitalization level disease. And I guess medically, one context to frame that in, I mean, there are very scientific studies about this, but medically, one context for that would be, obviously, there are many people on B-cell depletion therapies, right, for a variety of clinical reasons. And those people either can't make any neutralizing antibodies against the virus, or very low neutralizing antibodies against the virus. And yet, in general, they haven't had much increased risk of poor outcomes with COVID-19 compared to healthy people. Now, there certainly is some elevated risk, right? That's you definitely want to have those antibodies. But if it was all about those antibodies, those people would be at very high elevated risk, and they're not, suggesting that if they get infected, if they get a case, there are other components of the immune system that can serve as compensation for lack of a great neutralizing antibody. So that's the reason for attention to T cells. Plus the fact, in our original paper, we focused on T cells some because everybody was working on a spike vaccine, okay, in the absence of data. And we're like, look, that makes sense. But if it turns out that spike is a poor antigen for T cell epitopes, you have to have a T cell response to get an antibody response. If you don't have a CD4 T cell response, which is my lab's expertise, you can't get those neutralizing antibodies usually. And so if that's a problem, you really want to know that quickly. And what we instead saw was, 100% of people had good CD4 T cell responses, and spike was a very good T cell target. And so one of the things we said in that paper was, okay, that probably indicates that spike-only vaccines are actually a reasonable target. So that was one of the reasons to call on the T cells.
That gets us to the fact that T cells are very rarely measured. There's no commercial assay. Your group and La Jolla Immunology Institute have been leaders in this space. I think we've learned so much from you and the group. Of course, some other labs around the world, but one of the things that's comforting is with the variants like like Delta, the T cell responses are strong and seem to not be affected nearly as the neutralizing antibodies. And that, of course, you would think, oh, well, good, we won't have to worry about this. You would think, but then there's still obviously concurrently tremendous concern about widespread need for boosters. So if our on-demand system is so good on these reserves, T cells are not so much affected because of these diverse epitopes and the ability to deal with the changes, evolution of the virus. Why are we likely or possibly going to need a lot of boosters?
Yeah, great series of questions. I guess a couple quick answers. So yeah, there are multiple LJI studies on this general topic. So one recent one led by Alex Setti was that, yes, almost all the variants have there's negligible effect of T cell recognition of them, as far as I could tell. And in a preprint we put out just a couple weeks ago, I think we have the first report of T cell memory 6 months out from an RNA vaccine. And that's one of the things we haven't known about RNA vaccines, right, is how long do they generate memory for? And so that's a big question for when you might need boosters. And we did this for the low dose Moderna vaccine, the 25 microgram. And really, quite impressively, there was essentially no change in T cell memory between 1 month post-vaccination and 6 months post-vaccination. And so it looks like the RNA vaccines actually generate impressively stable T cell memory, which is a positive sign. then when you get to, okay, well, if these backup systems, so to speak, right, the the reserves, the military reserves are so good, why would you need boosters? And I'd say that comes down to two things. One, there's not proof that T cells are doing that protection. Okay, it's much harder to show scientific proof for T cells than antibodies, because antibodies you can just passively transfer, right? You can just purify them and transfer them into another person or a monkey and have a direct experimental proof. T cells, for one, are studied less. It's probably like 30 times more resource intensive to study a T cell response than an antibody response. It's just harder in tougher samples. And then biologically, it's tougher to prove. So you got to hedge somewhat there, right? Because there isn't a proof on that side. And then second, it comes down to one's definition of protection and what do you want, right? So one level of protection is at that hospitalization and death, sorry. And another level of protection is obviously infection and or symptomatic infection. And it's less likely that the T cells are contributing protection against symptomatic disease and certainly against infection, because they take some time to kick in. Right? They have to recognize that first of all, they have to wait until you're infected. Then they have to detect the infection. Then they have to get to the site of the infection and expand and whatnot. And normally that's gonna take time. So that amount of time is fine for prevention of hospitalization, but this is a virus that's really fast at replicating, again, right, in your upper respiratory tract and transmitting in just a handful of days. And so if the goal is instead to prevent symptomatic disease and prevent transmissions, you want to wait more, you want to bias things more towards that antibody component. Yeah.
One of the things that also comes up that you would you were one of the first groups to show the sanguine long-term protection, going out to 9 months. And the other thing is that's the prior COVID people. The issue here is that people that when they've had COVID, knowing confirmed COVID, they say, well, I'm good to go. I don't need a dose of a vaccine. But would you address that the vaccine would take that to yet another level? And is a prior COVID and one dose of mRNA better than even 2 doses of mRNA vaccine?
Last question first. Is prior COVID plus 1 dose of RNA better than, anything else? Yes. I summarized that literature in a 1-page piece in Science a few weeks ago, which I called hybrid immunity. And I think Eric, you and some others have called it superhuman immunity, which is or bulletproof, which is fair enough too. The immune responses in those individuals, the T cell and antibody responses in those individuals is dramatically higher than what you get from either infection or immunization. Do people with prior COVID need vaccination? Actually, I think it's a topic on which reasonable people can disagree, because the epidemiological data has been out at 8-9 months post-infection. Those people are still something like 93 to 100% protected, depending on the study, and those are relatively large studies. So that's a pretty good evidence. However, against variants, that protection drops, and how much that protection drops really has not been quantified well. The best paper on the topic is really the Science paper on the Manaus outbreak, which suggested something like a 30% drop in protective efficacy against the P1 variant compared to the parental in terms of reinfections. And that's obviously a big drop if that's occurring in other contexts and or other variants, whereas the vaccines seem to be more consistent than that. What we said in our Science paper, where we were measuring the immune responses, the immune memory in people who had COVID, was out at 8 months, it was quite encouraging that the status of the discourse before our paper was pretty centered on maybe people don't have any memory at all, and maybe people don't have any protective immunity, and maybe coronaviruses are just really strange in this way, right? And we didn't think that would be the case immunologically, but obviously data is what you want. And so, that was the largest ever study of immune memory to a virus over 6 months, with each of these different immunological compartments. And we showed people do have antibodies over that period, but a number of groups have shown that. We showed they have memory B cells over that period. If anything, they have more at 6 months than at 1 month, and that memory T cells were retained rather well over that time period, such that we projected that people who have had COVID are likely to have a reasonable amount of protective immunity for years into the future, i.e., that it wouldn't necessarily prevent infection or even necessarily symptomatic disease, but would prevent serious outcomes. And the caveat we put in that was one of the other, I think, major attribute of that data set was that there were 100-fold differences between individuals in those components of immunological memory. And we don't know which ones are most important over time. And so, if you show me 100 people who had COVID, some of those people are going to have very low immune memory, and I don't know which ones, and a serum antibody test can't tell me. And so, from a public health perspective, right, public health officials always want to lean on the safety side, right, include everybody's side, in which case the recommendation is certainly then all of those people should get vaccinated. But if they want to roll the dice and not get vaccinated, they probably are protected, most of them. That's where the data are. Sorry, that was a fairly long answer, but it's one of my favorite topics. So
no, it's a complex topic, and you're helping to dissect it and simplify it, Abraham.
Yeah, I want to just pick up on that. I think you really are remarkable in the way that you simplify and help the rest of us understand, both on Twitter, but as we talk to you, these difficult immunological concepts. And I can't let you go without asking you about your biography of David Baltimore, which I think most of our listeners are not aware that you wrote this wonderful biography ahead of the curve about a remarkable figure in science. And you're living through an era where multiple sort of scientific superstars, including my co-host, have emerged as faces of this pandemic. And are you tempted to write about this, to tell that story at some point?
That's a really good question. The answer is no, because writing a book really takes a lot of time and focus. And as an academic scientist, academic science jobs are 110% of your available time, right? If you're really trying to do cutting edge stuff, it's just not really compatible to do both research and writing is what I found. Certainly doing some communicating to non-scientists, right, via Twitter and other things, I'm super excited to do. I certainly tell people if there's just one person you follow on Twitter, it should be Eric Topol. I mean, in terms of COVID, it's really incredible how consistent and fast he is at finding the good stuff. In terms of writing a biography of David Baltimore, yeah, I'm certainly proud of the book. Plenty of people have told me they like it, and that it's good. So I wrote it as a book for non-scientists to help explain. I was trying to use the life of David Baltimore as a prism into molecular biology, RNA viruses, HIV, cancer, and immunology, because he worked on all of those topics. And so it was a fun book to write that way. And I looked around, and there were really few books written for non-scientists that were biographies of living scientists, and so that biographies can be pretty fun, right? But science moves pretty fast. So reading a biography of a scientist from 100 years ago just doesn't give you insights into the types of things that are going on nowadays. So that was the goal there. Yeah, thanks for bringing it up, Abraham.
in trying to wrap things up, we could
Sure.
talk to you all day, actually, but there's a couple of things I want to mention. One is, I think one of the reasons that you are so influential here in the pandemic, you in the group there, is that not only are you doing some unique work in immunology and, kind of a T cell capital, but you also are, because of your background, whether that was at MIT English communications, you're able to, come up with the ways to explain this stuff. And it is not easy at all. I mean, the way immunology is involved, I don't know any discipline that's become more challenging, arcane, and everything about it is a real challenge. But I also want to mention that on your wall is a picture of Tony Fauci holding up one of your papers. So can you just, as we finish up, can you tell us that story?
So I'll tell you, if it's a video interview or a video podcast, I don't use this seat because it's a little too obvious. But Tony Fauci held up our first COVID cell paper in Congress to try and use it to explain the importance of T cells to politicians. And you can see all our names on the paper when he's holding it up, so people loved it, right? Everybody at LJI was super excited about it. The president of LJI, Mitch Cronenberg, asked Tony Fauci if he'd be willing to sign a photo of it, and so he did. And that one's an autographed version of the photo. And so I said, well, what are we going to do with this? So I asked my lab manager where we should put it, and she said, we're putting it right there.
Well, it's actually remarkable because, as much as the pandemic has taken its toll on so many lives and morbidity, it's also brought out the science, the extraordinary science. And you've been a real leader in that. And to see a paper being shown in a congressional hearing, I don't remember that before or since. Okay. And
certainly not in a positive light.
Yeah, right. There you go. Anyway, so congratulations on that. We're really thrilled to have had a chance to discuss this with you. It's been a great educational session. Keep up the great work. We need you, and we need to learn more about this stuff because, this pandemic isn't over. We have other phases to get through, and there'll be more, of course, in the years to come. So, look, thanks for your extraordinary work, and we'll follow it with great interest.
I would say one reason for the group's success during COVID is Alex Setti, Daniela Weiskopf, and a whole bunch of people at LGI really have a long history of being good collaborators and easy people to trust, and so we were able to gather people together quickly and get people to trust us to, provide samples and whatnot. And to have Alex Setti in the building, who's one of the top couple of people in the world at identifying human T cell epitopes, which is no small puzzle in and of itself, right? Made it so we could really attack the thing full speed and just focus on trying to get to solutions, right? As opposed to worrying about a lot of other stuff. So yeah, it's been a real privilege as well. But yeah, thanks for the compliment.
And Shane, keep coming up with those post-it metaphors for mRNA and things like that.
Burglar alarms, fire engines, military.
Exactly.
That's how we can understand stuff. Thanks so much, Shane.
Thank you.
Thanks to you too. Have a good one, you guys.